working, so trying to work on that. So I'll try and push for a new sound. It takes some time to fix that. But what I was hoping is that we would alternate the podium from one side after each lecture. So one lecture there, one lecture here. So I think the representatives, so once we put the other podium, should instruct the instructor to alternate. This way it becomes fair for male and female students. Okay, I'll just finish the lecture from last week and then I'll start the new lecture. For Sunday's Mall, these lectures will not be going to be included. As you can see, there's theme. The Mall 2 will be all the lectures that I gave you, and I think one lecture from Dr. Raja. So that's how it's going to be, okay? Now, from last week, we were discussing the protein targeting, meaning protein is synthesized. What's next? Well, how do proteins, some proteins go to the nucleus, and how some proteins get excreted outside the cells, and how some proteins go to organelles? There's a signal. And if it's a protein that's going to be excreted, it has to go to Golgi apparatus, where it gets packaged and excreted. Can you settle down? Can you close the door, please? Just close the doors. No one is allowed. It's 5 minutes past the hour. The other door as well, please. No one is allowed after 5 minutes. And that's how it's going to be from now on. You already had 5 minutes before the end of the previous lecture, 5 minutes after. So that's 10 minutes. You got your 10 minutes. Fair is fair. So how do these proteins, synthesized protein, go to the place where they should be? In this case, we have the protein being synthesized, and this protein is targeted for excretion outside the cell. So you can see here we have what is known as signal peptide at the end terminal of the polypeptide being synthesized. Now this signal peptide, it's a signal that this protein is going to be excreted. It has to go to Golgi apparatus, get packaged and excreted. So you'll see another signal called signal recognition particle that binds to the signal peptide. And then you can see that the whole complex of ribosome and the synthesized peptide gets bound to the SRP receptor protein, which is the signal recognition protein receptor protein. You can see this signal receptor protein basically is hydrophobic, so it can go through the hydrophobic membrane, and you can see it going through this receptor, SRP receptor protein. And then what happens? It releases the signal recognition particle, SRP, and the polypeptide is basically being excreted inside the endoplasmic reticulum. Why do you think we call rough endoplasmic reticulum versus smooth endoplasmic reticulum? Rough because of these ribosomes attached to the endoplasmic reticulum. So what happens? You can see the signal, the green polypeptide fragment here gets chopped by an endonuclease. This is, I'm sorry, by proteinase. This is a protein, so it has to be proteinase. Proteinase, from the name, proteinase, breaks the proteins, chops it, releases the protein. Now the protein can be packaged by the Golgi apparatus and excreted outside the cell. What about membranes, nuclear envelope? I can see people still walking in. Let's say that we have a protein that's going to go to nuclear envelope. That protein, how does it know that it has to be localized in the nuclear envelope? Also, we have what is known as signal localization for nuclear envelope. And it's called, actually, more specifically, the nuclear localization signal, that is NLS. Nuclear localization signal. Basically, that signal tells the protein to go to the nuclear envelope and be part of the nuclear envelope. And this is applicable to all proteins going to different organelles. There is a localization signal so that the protein knows after it's being synthesized where to go. We do have co- and post-translational modifications. Co, while it's being synthesized, we have modification, translational, post-translational modifications. Co with, so while it's being synthesized, it's happening. Post, after the proteins being synthesized, get synthesized, the modification could happen. And we do have folding, protein folding. You know that we have for any protein, primary, secondary, tertiary, quaternary, we have folding, right? There are proteins called chaperone proteins. And these chaperone proteins would help the proteins to fold after they get synthesized. Of course, you'll see that there's a lot of sugars, phosphate groups, fatty acids, and many other molecules that get added to the protein. These are called post-translational modifications or co-translational modifications. Let's just take some examples. Some of these translational modification, post or co. So the first one is covalent attachment, where we add phosphate group. We already know there's kinase and phosphatase, right? Kinase adds phosphate. Phosphatase removes the phosphate group. So we do have phosphorylation, which could occur at serine residues, amino acid, threonine, or less frequently tyrosine. So you can see here that a phosphate group gets added here or here for the tyrosine amino acid. So what happens is that this could activate or inactivate the protein. We know that some kinases would activate, some phosphatase would inactivate, and vice versa. So these are covalent attachment where you add phosphate group. Other modifications would include addition of carbohydrate, glycosylation, basically, of polypeptides. And this carbohydrate chains could be attached to the hydroxyl groups of serine, this example, or hydroxylysine, and this is known as O-linked. You're going to hear these words when you study carbohydrate, O-linked. And the reason we say O-linked, you can see the linkage right there between the serine and the N-acetyl galactosamine, basically. You can see the O-linked group of carbohydrate attached to the serine. Could be also an amide group, and this happens with asparagine, where you can see an amide group that's linking between the acetyl glucose basically to asparagine. You can see the amide linkage right there. Okay, so this is a second modification, post-translational modification that could occur. Third one, which is very important, that is hydroxylation. Do you know scurvy? You remember the pirates. What happens with the pirates? They always have lost teeth, even though they show them looking cool and so on, but they're really in these sailors who used to sail for a long time. They usually have teeth loss, pale skin, sunken eyes, and so on. This is all because of vitamin C deficiency. Vitamin C is present in fresh fruits, right? So these don't have access to that during these long sails. So as a result, we have this kind of shade that happens to most sailors who sail for a long period of time. This used to be in the past. And this is because of ascorbic acid, which is vitamin C deficiency. We have an enzyme called hydroxylases. And this hydroxylase, basically, it adds hydroxyl group to alpha chain of collagen. The alpha chain of collagen gets hydroxylated, and we already know that collagen is a fibrous protein, and it's involved, basically, it plays a role in tendons, cartilages, and bones, as well as the cornea of the eye. So when there's no hydroxylation, there is no collagen. As a result, you end up with scurvy. Okay, this is post-translational modifications as well, just to show you how significant these modifications are. Finally, we have trimming. What is trimming? After the peptide or polypeptide is synthesized, it gets trimmed. One of the most important hormones that we have in our bodies that gets trimmed is insulin. Insulin gets trimmed where it gets split into C-peptide and the active insulin polypeptide. Another example, trypsinogen. So the proteins are excreted as inactive proenzymes. So trypsinogen, from the name trypsin, when it's a protease, it breaks down tissues, it breaks down cells. So we don't want to kill the cells, our own cells. So it gets excreted as inactive trypsinogen, and when it's needed, it gets chopped so that it becomes trypsin, it becomes active. And this is done by proteases. So this is an example of how proteins are synthesized as what is known as zymogens, proenzymes. Caspases, for instance, procaspase, it's inactive. And when the cell is about to commit suicide, the procaspases get converted into caspases, active caspases, to degrade the cells. These are just examples of enzyme to protect our own cells. Okay, with that, I'll conclude with last week's lecture, and I'll start today's session. So we know how transcription occurs. How do we regulate transcription? Is it under regulatory mechanisms, or it just happens all the time? Well, it depends, and we'll give some examples. These are the objectives of today's session. Basically, we do expect you to understand how eukaryotic gene expression is regulated at different levels, different stages, and we'll discuss the epigenetic inheritance, and we'll define what is epigenetic inheritance. This is a hot research area right now, because we have two alleles, one from the male, one from the female. Why is it? Usually they're both active, but sometimes we only have one and the other one is not active. What determines what's what? And when control of gene expression occurs not at the DNA sequence, this is what we refer to as epigenetic. And we'll discuss the definition of epigenetic inheritance. So we just described post-transcriptional or post-translational modifications. Now we're going to discuss regulation of this method, of these methods. And we'll discuss RNA interferences. If you are engaged in any research activities in a lab, basic science lab, most likely you will be working with RNAi to shut down certain protein expression and so on. So we'll discuss this as well. But at the same time, there's a hot research area right now, which is called microRNA. If I take plasma, this question I asked 20 years ago. I asked the students, if I take plasma, can I isolate RNA from the plasma? What do you think? Why? Where do you get the cells from? There's no cells, right? 20 years ago, the answer was, no, we don't have cells. But now, we have what is known as microRNA. You have more than 1,000 RNA synthesized by the cells, and it's in your plasma. And these are being utilized currently as markers for cancer, for diseases, for many things. We'll figure out what's their role. This is one of the hottest research areas right now, where we can characterize someone, predict if someone is going to develop diabetes, or predict if he's going to have cancer, certain cancer, by looking into these microRNAs. So the plasma, believe it or not, as I said, 20 years ago, we'd say no, but now, yes, I can isolate these microRNA from the plasma. So what makes cells different? Do you think your cells right now, whether they're in the nervous tissue, whether they are in kidney, in liver, do they all have the same DNA or not? Yes. How is it that we have different cell types? This is what we refer to as, we have what? Yes, this is what we refer to as differential gene expression. Differential gene expression. The cells become specialized based on some genes will be expressed, some will not, or are not going to be expressed. So how do we control this? That's what we're going to discuss. How do you make a cell specific for certain tissue, or specialized, what we refer to as specialized? And this is because of the differential gene expression. Now, we already know that we have DNA. This DNA becomes basically an RNA. It's capped, oligo, poly-A, basically is added, gets translocated to the cytoplasm for protein synthesis. Now, this RNA can get degraded or can be utilized for synthesis of polypeptide. And now we could either have an active protein or this protein could be degraded. The question I have for you, if I have an mRNA, how many times can we synthesize a protein from that mRNA, that single mRNA? 1, 2, 3, 4, what do you think? More than one? Yes, more than one. The mRNA could be utilized over and over and over. To shut down the protein synthesis, you gotta degrade it. That's why we have degradation. This is one key mechanism to shut down protein synthesis, is to degrade the mRNA. Now, we do have regulatory mechanisms at each step I just described. The chromatin, first, we have to unpack the chromatin. We have to loosen the chromatin so that the transcription factors could penetrate the chromatin, bind to the DNA, and express the mRNA. So there's unpacking, and this unpacking involves two mechanisms. That is histone acetylation and DNA demethylation. We're going to describe these two processes. Histone acetylation. So we have the histone, we're adding acetyl group to loosen the chromatin so that transcription factors could get in and bind. So we do have transcriptional control, conversion of DNA to primary RNA transcript, and then processing of RNA is also under control. The mRNA translocation into the cytoplasm is also now under control, even though it doesn't say control. I'm studying nuclear envelope proteins, lamins. Lamins control trafficking of mRNA in and out. And that's how they control, basically, conversion of mRNA into a protein by blocking or allowing certain mRNAs to go in and out of the nucleus. We have translational control as well to convert an mRNA into protein. We have stability issues. Now, here's a question for you. Let's see if you've done your homework. Eukaryotes versus prokaryotes, which one is more stable? Eukaryotes or prokaryotes? Eukaryotes, why? Because we have capping and we have poly A, right? Did you see in the prokaryotes if they have these two? Right? So we do have stability issues, and usually the stability comes actually from the 5' UTR. I'm sorry, from the 3' UTR, untranslated region. These are the kind of questions I would ask you if you really wanna, if I'm gonna be picky with you. Just telling you how you can link things together. So the stability is control mechanism. It could be degraded or it could become inactive. I mean, once it's unstable, it's inactive. And we do have post-translational control. Now, basically, when we say transcriptional control, all what it is basically to control when and how often a gene sequence is copied into mRNA. As simple as this. When and how. We do have two key mechanisms for transcriptional control. I'm not talking about translational controls and so on. I'm talking about transcriptional, a gene to be synthesized basically, or mRNA to be synthesized from a DNA. The first one, it is structural chemical modifications, which I just told you about. And that is acetylation of histones and demethylation of what is known as CpG nucleotides, cytosine, guanosine nucleotides. We call it CpG, C-phosphate, guanosine, CpG. You're going to hear this term, CpG islands. When you see a lot of CpG or CpG island in the promoter site of a gene, you know that that gene is basically shut down. It's inactive, as simple as this. And we'll talk about, this is in terms of methylation. When that promoter is methylated, basically that shuts down, turns off the gene expression. Another key transcriptional control mechanism is at the level of DNA binding proteins, and this is when we describe transcription factors, which could turn on or off the signal. So the first step of transcription involves basically unpacking of this highly packed DNA. So these packed heterochromatin basically are usually not expressed. We need to loosen them. So we have to modify these chromatin, packed, compact chromatin. We have to modify it chemically. We're going to discuss acetylation. And just to loosen the DNA so that mRNA is expressed. Thus, RNA polymerase would come and start the whole process. Now, the RNA polymerase complex has to be stabilized by transcription factors. At the same time, we have what is known as enhancers or activators and suppressors. We're going to describe key terms. That is confusing. I tried to make it as simple as possible for you in the next few slides. What is an enhancer element versus an activator or suppressor? So I tried to simplify it, make it as simple as possible, because these terms you're going to get in the exam, where we're going to ask you about certain enhancer elements versus activators and so on. So we do have transcription factors to stabilize the RNA polymerase complex so that RNA could start the synthesis. So let's start with the first mechanism, and that is chemical modifications. And that is histones are acetylated, basically. But before we talk about histone acetylation, let's describe histones again so that you could remember it. We already know that histones are composed of octameric disk that has H2A, H2B, H3, and H4, and we have two copies, and this is basically would form the nucleosome. We have the H1 linking, basically. This is the H1 outside the core, the histone core, and we have the DNA linking all these nucleosomes. This is the structure of histones. Now, with histone modifications, all what it is, you can see that we have compact heterochromatin material, and then we have histone tails, and basically this is where the histone gets acetylated. We're adding acetyl group at these tails. This would loosen the chromatin so the transcription factor could get in, start transcription. Here's my question to you. We have a group of histones called proteins, histone proteins, called histone deacetylase. And we have many of these histone deacetylases. Do you think that histone deacetylase, they remove the histone, make it compact? Do you think these proteins are aging or anti-aging proteins? What do you think? They're anti-aging. We have proteins called sirtuins. Sirtuins come from a protein called SIR2, silencing information regulator, silences the gene expression. SIR2 has been discovered as an anti-aging protein that could prolong the life of Drosophila, Candida albicans, and many other yeast, fruit flies, and so on. So the research now is ongoing. This is an active research area. Can we find the homologue in humans? Can we find something that's similar to the SIR2 that could prolong the life of humans? What is the only way you could prolong the life of a human being? Or let's say now we know that in mice, we can prolong the mouse life. What's the only way that's present right now? Caloric restriction. You restrict the diet of a mouse, you prolong its half-life up almost 50% extra. If you restrict his diet, and diet restriction would induce histone deacetylase sirtuins, as simple as this. I'm sorry? In humans, we have 7 sirtuins right now. We don't know which one is equivalent to this SIR2. Okay? So this is one active research area I'm doing right now. We just developed a method. But just to give you an example, why do you think that people say anti-aging is to drink wine every day? I'm not advocating wine drinking, but I'm saying what is being said. Why is it they say wine extends their life? There is an active substance called resveratrol. Resveratrol is in the red grapes, resveratrol. And that resveratrol, you can buy it. It's a supplement, you can buy it. And it's being sold as anti-aging supplement. It's called resveratrol. And this is what they say is the active substance because it induces SIRT1. Anyway, just want to discuss this because the fact it is related to histone acetylation, deacetylation. So we have enzymes that would acetylase and enzymes that would deacetylase, and you would expect that the histone deacetylases would keep the cells not expressing, they're not burning themselves by expressing too many genes. That's how you look at it, okay? So we do have histone acetylation. So acetyl, think of it bulk being added to the DNA so it opens up the chromatin. So transcription factor could walk in and start the transcription. That's how you look at it. You can see that this is unacetylated histones. The minute they get acetylated, they're loosened. Now the DNA polymerase or RNA polymerase in this case would get in and start the synthesis and transcription of mRNA. Another mechanism, DNA methylation, which plays a major role in genetic imprinting or inheritance. And what is methylation? Basically, it's adding a methyl group to the cytosine. I told you the CpG. island. So when the gene, look, you can see that this is the promoter right there. It has cytosine. When it's rich in CG, because double strands, that's why it's called CPG, because C pairs with G, right? Cytosine with guanosine. So when it's not methylated, the gene is active. When it's methylated, added methyl group is added to the C. Basically, this would shut down transcription. As simple as this. And this is where we introduce epigenetic inheritance. Epigenetic inheritance, and you'll hopefully, when you take genetics next semester, you'll come across the genomic imprinting. And I already talked to you about genetic imprinting. We have two alleles, one from the male, one from the female. Usually the two. This is one of the questions that we have seen, I get asked several times. Are the two strands active? Or just one? So you have to look into two strands versus two alleles. What do you think? Are the two alleles active? Differentiate. Alleles, one male, one female, versus the double strands. That's different. The double strand, you have only one side, could be the sense or anti-sense in terms of the positive or the negative strand. But it always, you have one strand only being utilized for transcription. What about the alleles? Usually they're both active, but sometimes, for whatever mechanism, you get only one allele that's active. What happened to the other allele? Usually it gets methylated, basically, and it's rendered inactive, as simple as this. So we do have this mechanism which could regulate the expression of either maternal or paternal alleles of certain genes. Now, epigenetic basically is when the chromatin modifications do not alter the DNA sequences. There's no change in the DNA sequences. We're adding a methyl group. We're adding acetyl group. As simple as this. We're not changing the DNA sequences. And this is why we have what is called differential gene expression. You'll see that some genes have methylation on the promoter site. So it shuts it down, and we end up with specialized cells because this is a key mechanism that would shut down gene expression, basically, and thus we would have cells producing certain proteins becoming very specific cells. Okay, another mechanism is transcription initiation, and that is for the RNA polymerase to bind and get stabilized by transcription factors. I thought that it's important to understand what is an operon. You're going to see a lot of time lac operon. If you were a biochemist, the first thing I would teach you lac operon, because we do a lot of experiments with the lac operon. What is an operon? An operon is a functioning unit of DNA that could contain a cluster of genes. Or in prokaryotes, we already know that we have polycistronic, right? Polycistronic, where this is an mRNA coding for multiple proteins. This is of course, they have one. What do you think here? We have one or two promoter sites for this polycistronic. Do we have multiple or one? We have one. This is called an operon. That promoter controls all these genes. That's why we used to say the operon is for prokaryotes, but now they found genes in eukaryotes that act as also an operon, multiple genes regulated by one promoter. This is what we call an operon. So transcription factors, we already hear about transcription factors. You need to know there are two types of transcription factors. General transcription factors that will induce gene expression. These are general. They're not specific. We have specific transcription factors that could be activators or repressors. Learn the terminology. Specific transcription factors could be activators or repressors, because you're going to hear about enhancers, about suppressors, and so on. Know the differences. Specific transcription factors are activators or repressors. So now we start into this. This slide has a lot of terms, many terms I want you to learn. You can see here we have an enhancer, and the more accurate term for it is enhancer element. And this is what is known as cis enhancer element. Cis, because you can see that it's in the DNA. It's a piece of DNA. And it's called enhancer. We always say enhancer. It's enhancer element. And it's regulatory element. It regulates transcription. We're going to see what's true. We have silencer element, which is also a cis element. It's a piece of DNA. It's part of the DNA. Then we have what is known as an insulator. And I'm sure that most of you, this is a new term for them because they're used to the silencer and enhancer elements. Insulator, you can see that it blocks an activator of transcription factor, specific transcription factor, from exerting its effect on the operator site. So this is what is known as the operator site, where basically the regulatory proteins could turn transcription off and on by binding to a site such as the silencer element or the enhancer element. And then this is the activator or repressor. Then it binds to the operator site to turn on the gene expression. So these activator or repressors, this is what we call as trans-acting factors. These are cis-acting elements: silencer element, enhancer element, insulator element. These are what is known as cis-acting elements. These specific transcription factors, activators or repressors, these are trans-acting factors. Are we clear what's the difference between an enhancer and a silencer, insulator versus specific transcription factors that includes the activators and the repressors? You should know this. Okay. The repressor. Okay, now look into the activator. Activator binds to the enhancer element. And this, basically, the activator now can attach or activate the operator site, which is a site basically that could bind. These are the activator and other transcription factors and start the transcription. If you look into the repressor, it binds to the silencer element. It binds to the operator site, but does not induce transcription. This is why it's called the repressor, because it does not induce. It basically prevents transcription from occurring. If you look into the insulator, it blocks the activator from binding to the operator site, so there's no transcription. So do we know what's the terminology here? I mean, I tried to simplify it as simple as possible. You're going to see enhancers and so on. Key things about enhancers, these are control elements. So the control elements includes the enhancers, silencers, and insulators. These are control elements. That's why we call enhancer element. We call it silencer element, insulator element. The B is a repressor. It binds to the silencer element. Then it binds to the operator site. As a result, this repressor will prevent the expression, transcription. The insulator, on the other hand, blocks the activator from exerting its activation of the operator site. As simple as this. It seals it, insulates it, so the activator cannot induce transcription. Okay? I know I'm running out of time. I'll try and finish on time, and I'll ask for another half hour during maybe next week or after that, so that I can finish today's lecture. I can't hear you. Can you go to the mic, please? The insulator, it prevents the interaction. I'm sorry? B, what about, oh, the silencer. It depends on the transcription factor, specific transcription factor. When it binds to it, basically this repressor binds to this silencer element, and this would bind to the operator site and prevent the transcription. It's called repressor. This is the function of that specific transcription factor. Again, general transcription factor, specific transcription factors. I cannot make it simpler than this in terms of the terminology and so on. If a repressor is binding to enhancer or repressors, sorry, I can't hear. Can you go to the mic, please? An activator binding to enhancer, what do you think? Here's a question for you. If an activator binds to enhancer, would that definitely is going to induce transcription? Is what? To a silencer? Activator to a silencer? It's not going to work. These are specific sequences. But here's my question. The binding of an activator to enhancer element always associated with transcription. Is this correct statement or false statement? You have the figure right there, tell me. That's false. Why? Because you could have an insulator that prevents it from basically binding to the operator site. Right? You can see here, it's bound, but the insulator blocked it from activation of transcription. Simple. So now, key differences between, I know that the textbook, see, this is where the confusion is. You see enhancers, that's it, it's enhancer element. It's part of the DNA, but they don't say enhancer element, they just say enhancer, silencer. You'll see it in many textbooks, references. So what is an enhancer and what is what we call promoter proximal elements? The reason I'm putting it, it's in your reference, in your textbooks and so on, so it's kind of confusing. Let's look into the promoter proximal elements. These are relatively short nucleotide, basically 15 to 30 base pairs, and they are usually located within the 200 base pairs upstream of the cap site. Is +1 the cap site or not? Well, if I ask you a question, +1 nucleotide here, is it the cap site, capping? The answer is yes. That's where transcription starts, right? And then you have the 5' UTR, right? So the capping is the +1, which is the site for the beginning or initiation of transcription. That's where capping. Okay, you see, I'm telling you right now, biochemistry, you cannot just take the slides. You have to read your textbook or reference book so that it explains to you these things. I cannot explain it to you. I have only around 50 minutes or less to explain it. And I cannot talk about the detail, but these are the kind of questions I would ask you to show that you understand rather than you memorize just the slides. Again and again, I told you, 60%, if you only memorize the slides, you are for 60% of the exam. As simple as this. For that exam. You want to improve it? 20% almost. If you do what? Read your textbook. So you're going for 80%. You go for all the exams, review exam questions, then you're going for 100% of that exam. As simple as this. Yes. An insulator in general is a repressor. Function-wise, yes. Yes, in general, it will suppress. It is specific for one activator. And you'll see why when we discuss these transcription factors. This is specific activators, we said. So they have certain structure. I'll conclude with this slide, take the attendance, but I'll ask for another half hour, as I said, to finish this lecture, because believe me, I can play with the questions for you. I mean, it's so easy for me to play with the questions here. So I want to make sure that you understand the concept. Students get confused between elements, enhancers, suppressors, insulators. Just want to make sure that you do get it. So I'll go through the slide and I'll stop. We have the promoter proximal element. So we said that this is within the 200 base pairs upstream of the cap site. From the name here, when I ask you the question, is the 5+1 is the cap site? The answer is yes. Then we have an enhancer element. Look into the terminology, even though it says here enhancers, that's enhancer elements. It's part of the DNA. It's cis-acting factors. So the enhancer elements, they are around 100 to 200 base pairs, could be upstream, downstream, even in the introns. And this is within around 50 kilobases, upstream, downstream of the cap site, or within an intron. What about the silencer? Is it the same? Silencing element? Yes. So it should be enhancer elements and silencing or insulator elements is the same. Okay? So they could be anywhere. They don't have to be 5' or upstream of the cap site. Now, the silencer element mostly is in the upstream of the cap site, mostly, but it can be present in the introns or in the enhanced or downstream. Initially, it was thought as always as upstream, but then they found some enhancers in the introns and downstream. What about the insulators? Cell debating, it's a new thing. They're finding not that many insulators have been discovered so far. Okay, with that, I'll conclude. Take that attendance because I know there's a session UPP.